All right, let's dive in. Today, we're going to crack the code of life itself. We're going to explore how the genetic instructions inside every living thing are written, how incredibly, shockingly easy it is to mess that message up, and the absolutely brilliant digital tools scientists built to read it anyway. So let me ask you this. Imagine you're reading a book, but a single misplaced letter makes every single word from that point on turn into complete gibberish. That is the exact high stakes problem our cells are dealing with every single second. So, how does this whole language of life even work? To really get why this is such a huge deal, we first need to understand the rules of the game. So let's take a look at the basic grammar of our genetic language. Okay, so think of your DNA as the ultimate instruction manual for life. A gene is just a single recipe in that manual, and it's written in a super simple four-letter alphabet: A, T, G, and C. These recipes tell our cells how to build proteins, which are basically the tiny machines that do, well, pretty much everything. Now, here's the trick. The cell doesn't read these letters one by one. No, it reads them in three-letter words called codons. Each codon is a really specific instruction, telling the cell which building block, an amino acid, to grab next for the protein it's putting together. And this is what it looks like when everything goes right. It's honestly beautiful. Just look at that normal translation. The cell's machinery reads ACG, and boom, it adds a threonine. Next, it reads AGG and grabs an arginine. Then ACU, another threonine. It's this perfect rhythmic process, and the result is a functional protein, made exactly the way the recipe intended. But what happens when that perfect rhythm is broken? Okay, let's get into this, because this is where one tiny, tiny error creates an absolute catastrophe. You see, as precise as this system is, it's also incredibly fragile. Just one little mistake, one letter getting inserted or deleted where it shouldn't be, and the whole process can come to a screeching halt. And this shows the disaster perfectly. See that single A that just got jammed in there? The cell's machinery has no idea it's a mistake. It just keeps on reading in its little groups of three. So instead of reading ACG, AGG, ACU, it now reads ACG, AGGA, CUG. The entire reading frame has shifted. Every single codon from that point forward is now totally wrong. And the consequence is just a complete mess. The cell starts grabbing all the wrong amino acids, building a garbled, useless protein. And what's even worse is that this new scrambled sequence often accidentally creates a stop codon. That's like a giant red alert telling the machinery to just give up, ending the recipe right then and there. It is a total system failure from one tiny typo. Now, this brings up a huge problem for scientists, right? If you find a new string of DNA and the reading frame is this fragile, how on earth can you know how to read it correctly? Exactly. How do you read a code if you don't even know where the first word begins? Do you start on the first letter? The second? The third? Guessing is not an option. You need a much, much better tool. And that's where BLAST comes in. This is one of the most important tools in all of bioinformatics. It's a whole suite of programs, but we're going to focus on one really clever one called BLASTX. Its whole job, as you can see here, is to take a string of DNA, a nucleotide sequence, and figure out what protein it's supposed to make. Think of BLASTX as a digital Rosetta Stone. It solves that whole "where do I start reading" problem with a really simple but powerful strategy. It doesn't guess. It just translates the DNA sequence in all three possible reading frames at the exact same time. So here's how it works. It's actually a really elegant, kind of brute force approach. First, it takes your one DNA strand and generates three different potential protein sequences from it. Second, it takes all three of those translations and blasts them against a global database of pretty much every protein ever discovered. And third, it just looks for a hit. This visualizes it perfectly. You've got our one DNA sequence on the left, and it becomes three potential proteins. Two of them are the result of the wrong reading frames. They're just random gibberish. They search the database, and they find nothing. But the third one, the one translated from the correct reading frame, gets a match. It finds its identical twin in the database, and the computer lights up. That's our answer. Now, this isn't just some neat computer trick. This capability fundamentally changes what scientists can discover about the entire biological world. And this is where it gets really, really powerful. Scientists can now go into literally any environment, a handful of soil, a drop of ocean water, or even the microbiome inside our own gut, and sequence all the DNA they find.
They get millions of unknown gene fragments, and BlastX is the key that unlocks what they all do.
And that's the real magic. Finding that match does so much more than just tell us the correct way to read the gene. If our unknown gene matches a known protein that digests sugar, well, we've just discovered a sugar-digesting gene. We've gone from a meaningless string of ATGCs to actually understanding its biological function.
A tool like this gives us the power to read the blueprint of any organism, even ones we've never seen before or can't grow in a lab. It just blows the door wide open to an entire hidden world of biology for us to explore. It really makes you wonder, with a universal translator for the code of life itself, what incredible secrets are we going to uncover next?